Hi, everybody. Welcome to the Frank and Lizzie show. Today, hi, Frank.
Hi, Lizzie.
We have the pleasure of actually interviewing two people on this interesting technology. And the first one is Dr. Robert Snyder, who's the Dean and Professor of Clinical Research at Barry University School of Podiatric Medicine. Hi, Rob. Welcome to the Frank and Lizzie show.
Hi, thank you for having me.
Yeah. Hi,
Yeah.
Robert.
How are you?
Great. Excited to learn about what you're going to talk about.
Thank you.
So, Rob, I've worked with you for almost 5 years now on a variety of other projects, and I know that you have told me about a blood clot technology for years now. So excited to get to interview about this. Can you tell me more about what this ActiGraft is and how it works?
So basically, it's a whole blood clot therapy that is applied topically to the wound. It's changed every week. From a mechanism of action standpoint, it could actually act as a drug delivery system, although that has not yet been studied. It has multiple growth factors, which is really very interesting because when we think of growth factors, we look at platelet-derived growth factors and VEGF, et cetera. But there are probably hundreds or even thousands of growth factors that are sitting in this blood clot, which are going to now bathe this wound. We also have a significant number of macrophages in whole blood. Macrophages have two stages, M1 and M2. M1 basically is inflammatory, and M2 is anti-inflammatory. So what we want to do is we want to convert the macrophage into something which is anti-inflammatory. We also want to be able to fight infection with the macrophages as well. Another important factor is the fact that we have something called the extracellular matrix, which is collagen, elastin, fibronectin. It's the largest structure in the skin. And unfortunately, what happens when you have a chronic wound is that area is completely destroyed. So the blood clot actually fills the hole and acts as a scaffold so that keratinocytes, those cells that are in the peri-wound and in the epidermis, can move across it. Also, there's a communication between the extracellular matrix and other cells in the wound, something called dynamic reciprocity. And if you don't have that extracellular matrix, then you don't have that communication and the wound will not heal. So this blood clot therapy actually fills that hole. And lastly, it can stimulate pluripotent stem cells. So you can recruit stem cells as well. So it does many, many things. And we've had some incredible results on a variety of wounds.
Well, you know what? You're talking about results. But what's unique about this is that you're harnessing the body's own properties, healing properties, to tailor an approach for patient care. But let's talk about research because everybody wants to know about evidence. Can you share with us the evidence that you have on the product?
So there are 16 papers that are currently available. The ones that I want to focus on, number one, would be the registry that we did, which is really real world evidence. We took all comers, we used all kinds of therapies, including the whole blood clot therapy. And we found that at the end of 12 weeks, 95% of patients had healed in a study that we did on 29 of those 150 patients. We also looked at a consensus document to see exactly where this product would fit in. And we found that it had a profound effect on wound bed preparation. It could be used in a whole variety of wounds. But what's really interesting for me is the fact that we found that it would cover vital structures like bone, tendon, and muscle. And
Really?
that's really very, very difficult in many cases to do.
And
So I'm
you said
sorry.
a wide variety of wound types, so not just limited to like the typical DFUs that we see in a lot of these research studies.
So in the registry, we used all comers. So we had all kinds of wounds. We had wounds which were venous in origin. We had traumatic wounds. We had wound dehiscence, etc. We then moved to the randomized controls trial, which was performed on 119 patients. Actually, it was 16 sites from all over the world. So it was a really very comprehensive study. And what we found, as an example, that when we looked at the intent to treat group, which is really the most important element of the study, we found 41% healing versus 15% healing. And there is something called an odds ratio. An odds ratio is basically looking at variables. It's basically a binary. It's yes, no, to see whether or not we have improvement in one section versus another. So we found that this product actually was 3 times more effective than standard of care.
Wow.
So the study really put a exclamation point, if you will, on how important this technology is.
Wow. And Robert, if we had to do an odd ratio between Lizzie and I, we score high.
You
Um,
absolutely do. Probably
but
a 25. Yeah.
this article is phenomenal. This research that you did, where can we find it? Is this, can you tell us more about it?
Well, the article is in Journal of Wound Care. It just came out, and it is a very detailed study. It goes into not only the study results and a little bit about, in this case, we studied only the diabetic foot.
Mhm.
So the concentration was on that ulcer group, but it shows exactly how the product is used, looked at the primary and secondary endpoints. Of course, the primary endpoint besides safety was complete reepithelialization, which, as I mentioned, was very profound. And I think it's a well-written article, and I think it's enjoyable to read. It's easy to read, easy to understand, and I think significant in its results.
Well, it's certainly something needed to get this kind of higher level data out there. And if there's been nothing short of learning that in the last year, given all of the regulatory changes and whatnot. So, but from like a clinician standpoint, how do well, in my mind, when you say blood clot, I think, well, the first phase is hemostasis, and I'm trying to get on to the next phase for healing. And I think about, I'm always wiping away blood clots from my wound bed because, I want to be able to apply a product. But I also think of how slimy a blood clot could be like, and how in a diabetic foot or like a weight-bearing surface area, or even just a dependent area, how that might be challenging to like adhere. So could you talk to us a little bit more? I know you have vast experience with this product around the application and what it looks like for a provider to apply it in terms of the amount of time it takes and things like that.
It's an interesting question, because when you think about blood, you think about wiping it away so you can put
Yeah.
something else on it.
Yeah.
What happens here is basically you're drawing 15 cc's of blood from an individual. You're mixing it with calcium gluconate and kaolin, and it's kind of put into a dome, if you will. It's injected into a dome, and you shake the dome. It takes about 5 minutes for that to happen. And
Uh-huh.
then what you do is you basically pop it out and you put it directly onto the wound. It
Oh,
almost looks like a hockey puck when you kind of imagine what it would look like. You can actually bend it. It's very, very malleable. You can cut it, actually. It does ooze. It does drain a bit. You can cut it if you have to. But I found, and I've put on over 100 of these, I found that you didn't really have to do that. You could just take the entire clot and put the entire clot directly onto the wound and then fixate it, because it is on a piece of gauze. You can fixate it with steri strips. So I had an interesting experience. The first time I put it was on a heel wound, and of course the heel takes the most pounding, the most amount of pressure when you are walking. So the first time I put it on, I said, "Oh my goodness, I'm going to put this on. The patient's going to take their first step. The whole thing's going to squish and it's going to disappear." So what happened was something very, very different. Number one, when I took the dressing off a week later, it had completely disappeared. So you saw the remnants to the peri wound. But what I did find very interesting was one of the concerns I had was maceration, because
yeah.
again, I covered the entire surface, including the peri wound, and I was concerned that perhaps they would be, because of stimulation of healing, there would be a lot of maceration. So there was drainage coming out, but there was a little hole that nature created in the clot itself, so that the fluid actually came out of that hole into the dressing. So there was no maceration at all. And the wound was dramatically improved even within the first week.
So the
But
clot, doesn't occlude the surface of the wound bed. It does allow some transfer of fluids between the two.
Absolutely. I think that's the key.
Yeah.
There is no concrete proof of what I'm going to say right now, but my belief is that the wound actually signals the clot to give it what it needs when it needs it.
Yeah.
And there is that transfer of fluid. And something else to keep in mind, this is a scaffold. So not only is it replacing the extracellular matrix, but it's allowing the keratinocytes, those cells that are in the epidermis, it allows them to move through the clot. And if you look at an electron microscopy, you can actually see those cells in the scaffold. So it's really very, very interesting. So while that clot is working and dumping growth factors and doing all the things that it does, it's also allowing for those keratinocytes to move across. And if the wound is deep enough, probably fibroblasts as well. So you're getting the benefit of all of this taking place. Something else to keep in mind, the wound is actually not only contracting, but it's sealing itself off. The clot actually seals the wound off. So we now know, due to some new technologies, that a lot of bacteria that we see are not necessarily only in the wound, they're in the peri-wound as well. So what this product does is it seals the wound off so that bacteria can't penetrate, can't get in. So that's another reason why these wounds very often do not get infected.
Yeah. And I do have a question for you. As you know, we're going to have the person I consider the expert when it comes to reimbursement coding. But, you mentioned you use it all. So talk about wastage for us, because again, I don't deal with reimbursement, but I always hear about you have to document waste.
I don't think there is any waste here. I mean, you're basically drawing blood from the patient. So it's not like a product that has to be made or produced and then sent to you, or a product that's lipolized. You're basically taking the patient's blood. So the only thing that you're really wasting, if you can call it waste, is there's a little bit of blood at the periphery of the wound that's not actually in the wound itself. But my feeling is that when you're putting it around the surface of the wound, those macrophages are actually working on that peri-wound bacteria as well. So I don't believe that in this particular case there is waste at all.
So nice to hear from an expert like you. And of course, so nice to have you on the show.
Thank you.
I think we're going to speak to our next guest a little bit more about the reimbursement.
Yes. And thank you. And Rob, just to let you know, Lizzie and I are both a fan of you. So
Well,
thank you.
I'm a fan of yours as well. So thank you very much. Thank you for the opportunity.
And we're back with our second guest, and I have the honor of introducing Kathleen Shum. Kathleen Shum, she's the president of Kathleen D. Shum and Associates. It's a consulting firm that gives advice, strategic reimbursement advice to companies, manufacturers, and providers. The one thing I love about Kathleen is that she keeps up with the ever-changing reimbursement landscape. The other thing that I found interesting is that I didn't realize that you were rated in the world, and you're number 28 in the world in your specialty.
Oh my gosh, congratulations, Kathleen.
Thank you.
Such an honor to have you here. So Dr. Snyder gave us a lot of clinical information and mechanism of action and kind of why ActiGraft is so great. But obviously, coming from health systems and post-COVID, we know it's really all about sometimes the dollars and cents. And so our C-suite always wants to know about, how do we get this product in and get it covered? And so I just want to ask you a little bit more about, in terms of the coding and coverage for ActiGraft, what should clinicians or administrators know about the coding and coverage?
Well, I think it's very important to know that there for a while, we did not have coverage for autologous platelet-rich plasma. And that's probably because clinical trials weren't done or weren't done correctly.
Okay.
And then these new manufacturers who really wanted to bring this forward, they had to overcome a national non-coverage determination.
Big red flag, yeah.
And so in order to do that, their hurdle was higher because there was a national non-coverage determination. So they worked with CMS to do what's called coverage with evidence development. And they actually partnered together to do clinical trial in order to get CMS to understand that they should be covered. And when the coverage with evidence trial was over, then indeed CMS changed the national non-coverage determination to a national coverage determination. And I have to give kudos to the manufacturers who participated in that, because now we know that so much is about evidence.
Yeah.
And this particular category of products now has a national coverage determination, which means that all the Medicare contractors must cover it because it met the criteria for coverage development and a positive coverage determination. And so now if you choose to use this category of products, then indeed you know that it has to be covered.
And that's wonderful.
Yeah, I mean, that's the first hurdle, certainly. That was a big hurdle for them to tackle. And in terms of what they need to code, you know, is there a certain code that they need to use or restrictions in how many or sites of care, any of that nuances?
There's a lot. So the first step, of course, was just to get a coverage determination. But in a national coverage determination, there are many rules. But at least you know what the rules are.
Yeah.
And at the beginning, there was only one code that represented this category of products. And I hope all the listeners will listen very carefully to this because it's something they should check in their charge masters. Originally, the code that was out there for this category was G0460. And that category now means it is for ulcers that are not diabetic. And therefore, if you have G0460 in your charging system, you would be saying, oh, we're putting this on a non-diabetic wound, and therefore it would not be covered.
Oh.
So very important
Yes.
that everybody know the code is G0465.
465. I got it.
G0465. And everybody should check their charge masters because they may think it's in there, but it might be the old code.
Okay.
So we have a new code, G0465, and that means the national coverage determination only guarantees coverage for diabetic ulcers. And that code reflects that.
Okay.
In addition, it allows coverage up to 20 weeks.
Wow.
Which is quite nice.
Yeah.
However, you have to prove medical necessity for every one of the weeks. You can't just say,
makes sense.
you can't just say, oh, we're going to do it 20 weeks.
Right.
But the other nice thing about this is that if for any reason the ulcer is healing very well and you got to the 20 weeks and you needed maybe one or two more and you had the documentation that showed we've progressed greatly, but we're not quite there. The coverage determination also gives us the opportunity to have additional weeks covered, but if we do that, we have to add a modifier onto the code, which would be KX. And KX means we've followed the policy. We know we only have had 20 weeks, but we need a few more. And so on every application after 20 weeks, you would have to use G0465 KX. And your documentation would have to be addressing why do I need that additional application.
Medical necessity. Yeah, for sure.
Yeah, well, I tell you what, as we're wrapping this up, I really appreciate this information. I always stayed away. I went to one of your classes early on and I realized there's too much for me to know. I'm going to concentrate on clinical. So I do appreciate you enlightening us on these changes that impact what we do.
For sure. Thank you so much.
Well, thank you, and thank you for inviting me. It's great to be here, and it's great to be reimbursement on the Frank and Lizzie show.
Oh, this is another first because I love Kathleen. Now, I do want to highlight that, we heard from Dr. Snyder, and he talked to us about this product that uses the patient's blood, the patient's natural ability to heal, so we can change the environment. And now we heard from Kathleen Shum on the reimbursement, actually on the coding. So this is great. Thank you so much, Kathleen, for being here with us.
You're quite welcome. It's my honor to be here.
And thank you to our audience for listening. Be sure to like, follow, and subscribe the Frank and Lizzie Show on YouTube for more updates from these experts.